It's my great pleasure to welcome you to this week's genetics seminar. And it's our distinct pleasure to have Dr. David Pellman to be our speaker today. David has done amazing work scientifically, and I've been admiring his science for quite a number of years. So David graduated from college and as well as obtained his bachelor's degree as well as his medical degree from University of Chicago. Then he went to Boston and did his medical intern resident and as well as fellow in the Children's Hospital and as well as Dana-Farber Cancer Institute in Boston. He then went to Whitehead Institute and did his postdoc fellow with Dr. Gerald Fink. Then after that, he established his own laboratory in I believe in Dana-Farber Cancer Institute as well as Harvard Medical School from 1995. He has remained there since, and he's now the Margaret M. Dyson Professor of Pediatric Oncology at Dana-Farber Cancer Institute, as well as Professor of Cell Biology in Harvard Medical School. He also now have an important administrative role, and he's now the Associate Director for Basic Research at Dana-Farber. Dr. Pellman has made a number of very important contributions to science, and that really ranges from basic science, including going from the mechanism of spindle as well as cell division during mitosis, to very important questions associated with the genome stability that has fascinated both the cancer world and the disease world for quite a number of years. I remember when I was a PhD student, there was an argument in the field on what is more important for cancer. Is that mutations in tumor suppressors or oncogenes, or the genome instability that's caused actually many times associated with the cancer formation process that's actually more important in driving this process. And Dr. Pellman's work basically actually done one of the first work to demonstrate that amplification of the genome can actually accelerate the acquisition of mutations and genome evolution in this process. He has also characterized and the mechanisms for a phenomenon called Chromothripsis, which is a process by which many mutations can be acquired by cells very quickly to shape the evolution of the genome. Due to his amazing work, and many of them I really read with great interest and also admiration, he has been awarded many awards. It's probably going to be very long if I have to read all of them, but just want to point out that he has been a Howard Hughes investigator since 2008, and he was inducted as a fellow of the American Association for Advancement of Science in 2013. Without further ado, David?
Okay, thank you so much, June, for a very generous introduction. And it's great to meet everybody at Yale. I have a lot of old friends there, and I wish I could be there in person, but I'm very pleased nonetheless to at least have the opportunity to share some data with you. Okay, so this slide illustrates the general question that my lab has been after for a long time, which is to try to understand how you go from a nice normal genome with 46 chromosomes shown here on the left to an incredibly complicated cancer genome that can have hundreds, thousands, or even tens of thousands of chromosome rearrangements. And we've approached this, I'm sort of have bifurcated interests in the cytoskeleton and the mechanism of cell division. And we've kind of taken a bit of a, you'll see a cytoskeletal slant, if you will, on this question. This transformation from normal to cancer, we actually thought we knew how it worked in some ways. The prevailing model has been is that cancer genomes evolve slowly and gradually in the way that Darwin postulated normal organismal evolution would occur, so that you would get small scale genetic changes one after the other building up in sequential generations, taking you from normal to highly abnormal. But if you sit back and think about the numbers of rearrangements that you're confronted with in an actual cancer genome, you could really start to wonder how long this would really take if you went slowly and steadily one step at a time. And that's led some, including my lab, to start to wonder whether instead of going one step at a time, whether you could have massive bursts of events that would give you showers of mutations, and then small numbers of these events could more readily get you from normal to cancer and explain that highly complex genome in a more parsimonious way. And that I'm going to talk about, my group has been trying to understand mechanisms for these catastrophic mutational processes, and that's going to be the main subject of my talk today. One of the main themes that's emerged from this work is that the underlying events that lead to these catastrophic changes in the genome are linked to abnormalities in the architecture and the integrity of the cell's nucleus. You're looking over here at the left on a very extreme example of so-called nuclear atypia in a cancer cell line, and you can see all sorts of funny shapes of the nucleus, structures that I'll talk more about here called micronuclei, as well as structures like this called chromosome bridges. Sort of the structure of the talk today is I'm going to review some of our work on how these catastrophic mutational processes occur and what happens in structures called micronuclei. And then I want to address whether all these different funny shapes of the nucleus are doing different or similar things in terms of compromising the integrity of the genome. And I'm going to review some work that I'll propose to you points to a more unifying idea of how nuclear architecture impacts the integrity of the chromosomes. Okay, so we know of at least three major processes that drive very rapid evolution of genomes. The first I'm not going to discuss in great detail today, but it's something we worked on for a long time and grew out of our interest on the actin-driven process of cytokinesis. And that's the consequences duplication of the whole genome, which we showed back in 2005 here, could drive tumor genesis in a model in the mouse. And for many years, we worked on various aspects of the mechanistic events that lead to tumor genesis after whole genome duplication. This wasn't known at the time, but extensive whole genome sequencing of cancers has now revealed that something like 40% of all human cancers have undergone one doubling of the genome at some point during their evolution. The second major process that we're going to talk about here is called Chromothripsis. That's a Greek neologism for chromosome shredding that I'm going to have a lot more to say about. And the final topic are so-called chromosome breakage-fusion-bridge cycles, which I'm going to show a variety of ways in which this can lead to this phenomenon of chromothripsis, integrating what happens in nuclear structures called micronuclei and chromosome bridges. Okay, so what is chromothripsis? This was first discovered by Peter Campbell from whole genome sequencing in cancer, and I would say that it was one of the most interesting, at least to me, and surprising findings to come out of whole genome sequencing of cancer genomes. And what it was massive chromosome rearrangement of only one or a few chromosomes that was observed initially in a patient sample with chronic lymphocytic leukemia. Soon after Campbell's reports, numerous other groups reported the finding of Chromothripsis in cancer, and also several groups, including James Lupsky and Wicker Klusterman, described it as a cause of rare human congenital diseases for which there were not known mutations. Like whole genome duplication, Chromothripsis we now know is common in cancer. Somewhere around 30% of epithelial tumors have examples of Chromothripsis, and there are certain tumors like human pancreatic cancer that have extraordinarily high frequencies of Chromothripsis. So in human pancreatic cancer, about 65% of the cases have Chromothripsis, typically more than one chromothriptic event, which then add up to explain something like 50% of all of the genomic rearrangements in these tumors. We really don't know why these events happen frequently in some tumors rather than others, and that's an interesting question that my lab is pursuing in experiments I won't have time to discuss. But Chromothripsis, I told you, is not just specific to humans, and it's not specific to cancer. As I said, it causes rare congenital disease, but can also occur in plants. This was lovely work from Simon Chen and Luca Comai out at UC Davis, and that occurs almost certainly by very similar mechanisms that I'm going to describe to you today. So these events are genomic events that in principle can only be detected by whole genome sequencing. So although we postulated that the fundamental cause were cell biology events such as the missegregation of chromosomes, we were a little bit stuck initially on how to really nail down what was going on. And to address that, a lot of the experiments I'm going to talk to you about today involve a general approach that we playfully call Look-Seq, which would be a combination of cell biology approaches, cell manipulations, long-term live cell imaging, with the ability to then come into long-term cultures with a robotically controlled microcapillary, see events, grab the cells that underwent those events of interest, and then be able to perform single-cell whole genome sequencing on those individual cells at very high depth so that we can really understand a lot about the structure of their genome. And back in 2012, we had a proposal that Chromothripsis might be caused by chromosome missegregation events that led to the formation of these structures called micronuclei. And based on work from others, Emily Hatch and Martin Hetzer, we knew that the disruption of the nuclear envelope, the spontaneous breakage of the nuclear envelope, would somehow lead to DNA damage. And the hypothesis then was that these damaged chromosomes would be the source of Chromothripsis. And of course, what better way to get heavily isolated DNA damage than leaving most of the chromosomes intact, which is to partition some of the chromosomes into their own separate but highly defective nuclear structure. So we did experiments then where we made these structures de novo, watched the envelope break as a surrogate for the acquisition of DNA damage, watched the damaged chromosome come back into a functional daughter cell nucleus where we presumed it would be repaired by end joining processes, came in with our microcapillary, grabbed each of the cells and sequenced them separately. So this work is published, and I'm just going to show you a little movie that summarizes this published work. So you can get micronuclei from a variety of mitotic errors, for example, if you have too many centrosomes. And this leads to misattachments of chromosomes to the spindle, so-called near-telic attachments, which then cause chromosomes to lag at anaphase. And then the micronucleus forms because nuclear envelope assembly goes on the lagging chromosome. The same thing can happen if you have a single break that creates an acentric fragment of the chromosome that then also partitions into a micronucleus. These structures have few nuclear pores, bad nuclear import, undergo bad DNA replication. The envelope is fragile and ruptures, and then by unknown mechanisms, the exposure of the chromosome to the cytoplasm leads to DNA fragmentation. And if some fragments partition into one daughter and others go to the others, you get a reciprocal copy number pattern where the fragments, some fragments are in one daughter but missing in the other, and you get this mirror image for the affected chromosome, and then the fragments are all randomly ligaged together to create essentially a completely novel chromosome out of the starting chromosome with multiple mutations all happening simultaneously. Okay, so that summarizes some of what we know about the mechanism of Chromothripsis. And now I want to turn to another phenomenon, which is also a major feature and major cause of genome instability in cancer, called chromosome breakage-fusion-bridge cycles. Unlike Chromothripsis, we've known about chromosome breakage-fusion-bridge cycles for a long time from elegant experiments, genetic experiments that were performed by Barber and McClintock and Mays. And this is the scheme from a doctored scheme from McClintock's 1941 paper, where she proposed that these breakage-fusion-bridge cycles could be a source of gene amplification. And here's how the scheme goes. So her thought was that if the end of chromosome got broken off, in her experiments she used radiation. We now think that in developing tumor cells, this mainly happens by critical shortening of telomeres. So that you wind up with a chromosome, here's one arm, here's the centromere, here's the other arm, that lacks the terminal segment. This essentially looks like a double-stranded break. If you replicate that chromosome and the breaks are still there, you'll have two breaks next to each other that then fuse. And then the mitotic apparatus will grab the two centromeres of the resulting dicentric chromosome and stretch it into a bridge between the two daughter cells. And then, of course, once the chromosomes have fused, there's no memory of the original fusion site. So any random breakage is then going to give you a, so that you'll, for example, wind up with one daughter cell now here that has two copies of the A sequence and lacks the, and another daughter that lacks the entire A sequence. And then I hope you can appreciate that if you then iterate cycles of this, you'll get exponential amplification of this A terminal segment going from 2 to 4 to 8, etc. And can readily get heavy amplification of these terminal sequences. This gene amplification by the breakage-fusion-bridge cycles is widely thought to be one of the major mechanisms for oncogene amplification in cancer. However, despite the fact that we've known about these cycles for a long time, we've inferred their important biological significance, what's been much less clear are the underlying mechanisms driving key steps in this process. And in particular, questions like how is the bridge broken? Furthermore, although this process is thought to be very important in cancer, if you look in actual cancer genomes, you don't really see the simple pattern of gene amplification that's predicted by the BFB model in isolation. Rather, you can see some features of the cycles that are interspersed with much more complex rearrangements. So that raises an interesting question. Do we not see the simple pattern because it occurs, but then is obscured by subsequent events that take the simple pattern and rearrange it? Or is it that the original process is actually far more complicated than we originally envisioned? And I'm going to show you some data that supports that latter possibility. Okay, so one clue that that might be the case came from work, again, from Peter Campbell and his collaborator, Christine Harrison, where it was observed, at least in some contexts, that Chromothripsis and breakage-fusion-bridge cycles can co-occur in the genome. Whether that's a general rule or whether there was a mechanistic linkage was not clear. But that idea was also validated by some in vitro experiments by Titia DeLonga and Peter Campbell. However, and this I won't have time to go into this, there was a proposal that a nuclease that lives in the cytoplasm called TREX1 might be a critical factor in breaking the bridges and initiating the chromosome breakage-fusion-bridge cycles. However, our experiments trying to recapitulate that in similar or the same cells were not successful, and we then turned to look for alternative or perhaps additional mechanisms that could explain this phenomenon. And fundamentally, we're cell biologists and like to look at cells. And we started looking at the cells that had formed chromosome bridges. And we noticed that the bridges can get remarkably long. In a motile cell type like RPE1 cells, the bridges can extend up to 400 or 500 microns in length. And that got my postdoc, Neil Umbright, wondering whether the extension of the bridges might have something to do with their breakage. Here we're visualizing the bridges with a fluorescent version of a DNA binding protein called the barrier to autointegration factor. Okay, so to test this idea, Neil set up an experiment where he used soft lithography techniques to essentially paint a little corral of fibronectin into a micro pattern, which the border is shown here. And he made a simple long rectangle and compared it to a short rectangle. And in the long rectangles, the bridges, the cells can divide here, and the daughter cells will walk away from each other and extend the bridge, and you can see the bridges extending and breaking, and that looks like what would happen if the cells are unconstrained on a glass slide. However, on short patterns that limited the migration of the cells, we saw something different, which is that the bridges formed and they extended as much as they could, but they essentially never break. You can see this cell here went into mitosis. So this suggested that there possibly could be a mechanical component to the bridge breakage phenomenon that hadn't been previously appreciated. And that was supported by a little further imaging. What you can see here is, again, the DNA is shown here in green, and then you're going to see just really a weak actin stain that'll show you cytoplasm. What you're going to see here as we watch the bridges break, you'll see two things. First of all, you can see that the bridges can form acute angle bends here. And what you'll see is some cytoplasm that will, a protrusion of cytoplasm that will extend along the bridge and appear to kind of grab the bridge here and bring it back here so that this segment goes taut and this goes slack. Okay, here's the protrusion. This is taut. This is now going slack. The bridge will break. You get ruffling of the membranes, which is consistent with an abrupt loss of tension. So this doesn't prove that the bridges are broken with mechanical force. It's correlation, but it does raise the possibility that there's a mechanical component. And to address that, Neil did the following experiment where he induced the formation of bridges, allowed the cells to divide and watch the bridges form. And then he added inhibitors of either myosin contractility or a small molecule that induces the disassembly of actin, and asked whether actomyosin is actually required for the breakage of the chromosome bridges. And you can see here that there was a very strong result. Actin and to a lesser degree myosin contractility are significantly required for the breakage of bridges. Okay, this is good. It suggests that the actin and myosin have an interesting role and can actually promote mutagenesis by inducing, by participating in the generation of double-stranded breaks. It raises the possibility that mechanical force might be sufficient, but it doesn't prove it. This is an experiment that just shows a necessity, and we don't exclude the possibility that there could additionally be nucleases playing a role in this process. But those nucleases have to require stretching of chromosomes by actomyosin contractility. Okay, so next we wanted to establish both the immediate and long-term consequences of the breakage of chromosome bridges. And to address that, we used our combination of imaging and single-cell sequencing. And the first question that I'm going to address is, what are the very initial problems that the chromosome within this abnormal nuclear structure faces? Which I'm going to propose to you is a defect in the replication of the DNA within this bridge. Actually, before I show that, I'm going to look first at some of the consequences of mechanical breakage of the bridge. Okay. So in the very simplest kind of model, if the breakage is purely mechanical, you might think that there would be one DNA double-stranded break, and you would be able to see this would be a sister chromatid that's fused at the end. You get a break here, and in one daughter cell, you would get a terminal segment gain of one homolog. And I'm not showing you any of the details here, but one of the tricks that made the genomic analysis convincing is that we were able to distinguish the haplotypes of each of the homologs and look only at the copy number and rearrangements on the chromatid that was in the bridge. So we were able to detect bridge breakage events with one daughter gaining a terminal segment here and the other losing it in 20 out of 20 of the examples that we sequenced. And in a number of cases, they were simple breaks consistent with a mechanical breakage model. However, we actually rather frequently, more frequently, saw that the breaks were mildly complex and that you would have a small handful of breaks like this, where you have a fragment like this that was then rearranged. The green links here show the aberrant ligations of the ends of these DNA double-stranded breaks. And here you're looking at one daughter and comparing it to the other. Again, you have a mirror image, which supports the idea that this is going on. So one thing that immediately jumped out is that there have been huge studies doing comprehensive cancer genome sequencing that were recently published, so-called pan-cancer analysis of whole genomes. And this type of pattern was actually found all over the place in cancer genomes and was termed local jumps or local end jumps, and was noted, but there was no clear mechanism. But this, our finding here shows that breakage of bridges is one likely source of these of this pattern commonly now found in cancer genomes. But that doesn't say, you know, we don't know what is this purely due to breakage. Again, it's not possible to completely establish that, but one question that immediately comes up is that can mechanical force create these kind of ragged ends with multiple breaks? And to do that, what we did was to come in here with our microcapillary. Here's two cells that have divided and are connected by a bridge. And what we're going to do is that we're going to come in with fluorescence here. You can see the capillary stretching the bridge then breaks. And now we can come in and grab both of these cells and sequence them. And essentially what we found is that, again, we recapitulated all of the sequence features that we found with spontaneous bridge breakage. We could see some clean breaks. We could see some more complex breaks with these so-called local jump features. So we think mechanical force is an important factor in the chromosome bridge breakage fusion bridge process that's playing a role in shaping the architecture of these genomes. So that accounts for about 75% of the initial breakage events, but that still left us with about 25% of the cell pairs where we saw extensive rearrangement of the chromosomes. And what was responsible for that? So here I'm just going to show you a model, and then I'll show you a little bit of data to support that model. So we previously had shown that micronuclei after chromosome segregation are born with few nuclear pores, leading to defects in accumulating a lot of proteins required for normal DNA replication and repair. We wondered, and that causes a very striking defect in DNA replication. In a paper that we published in 2018, we showed that if we could correct the nuclear pore assembly defect of these structures, we would reverse the defect in DNA replication. And I'm going to show you an image on the next slide that shows that chromosome bridges have the very same defect in nuclear envelope assembly. So that made us wonder whether these structures, although they look, you know, obviously very different, whether they actually share a lot of similar functional properties. Okay, so this is just an image from that 2018 paper. Here's a lagging chromosome. You can see that some nuclear envelope proteins are assembling normally on the lagging chromosome, but nuclear pores are really strongly deficient in assembling on these structures, leading to the defects that I mentioned in the last slide. And you can see here that the same thing is happening for the chromosome bridges. So we address this in a variety of ways. In this experiment, we created cells with chromosome bridges. We pulse labeled them with EDU to assess whether you could see DNA replication in these structures. And you can see that the EDU is markedly deficient in the chromosome bridges. And this is not just because the bridges are very thin. We had controls to show that we could readily detect EDU within bridges if it were to occur. And then we could also confirm the defect in DNA synthesis directly by DNA sequencing. So why would we care about defective DNA replication in these either micronuclei or bridge structures? And this is just a slide to remember that when Chromothripsis was first discovered, there was quite a debate about what the underlying mechanism was. So one mechanism proposed was that the chromosome would literally shatter and the random bits would be ligated together. We think that our single cell analysis from 2015 established that clearly. However, there was a whole nother theory that you could get the same pattern from essentially rogue DNA replication forks. That's illustrated here, that you would have a DNA break and the polymerase would now use little bits, small bits of homology to start jumping around the affected chromosome and copying little bits from here, from the blue segment to the green segment, et cetera, et cetera. And you could see how defective DNA replication like this could in principle lead to a very similar pattern as the fragmentation and religation of segments of that chromosome. One thing that always comes up in major scientific debates is that oftentimes you can get vigorous disagreements, but the models being debated are actually not mutually exclusive. So in fact, you could have an abnormal nuclear structure that both predisposes to bad DNA replication and to chromosome fragmentation. And indeed, in our original paper on this, we saw an example where there were short sequences from all over the affected chromosome, like only 200-300 bases long. They're all assembled in tandem, really evocative of this kind of replication template switching type of mechanism. Within the larger rearrangements that we could establish were due to fragmentation and religation. So we proposed that both of these things might be able to go on in the same cell. And indeed, we have extensive evidence now for this kind of thing happening in multiple samples in chromosome bridges. And I'm just gonna, there are a lot of interesting features of this, but for the sake of time, I'm not going to go through it in detail. But there's a couple of points. So this is a series of chromothriptic rearrangements. These are the links of the abnormal joins. And we normally would think of that being random fragmentation and random rearrangements. But you can see here is that the breakage points are not random. There are in fact hotspots, and you can see that they're weaving back and forth. So this is the resulting chromosome with here's some of the green segment here connected to red segment. And there's some very interesting features. I want you to see that within a hotspot, there are segments of this hotspot that appear to be used only once. And it seems that bits of this hotspot are being sampled in a way that there's partial overlap between these segments. It's a very unusual and interesting pattern, but we've seen it now in numerous samples. And indeed, similar things have been reported in the pancreas analysis of whole genomes. And in fact, we were able to see this through a collaboration with Peter Campbell's group in chromosome rearrangements associated with Chromothripsis in a primary patient sample from renal cell carcinoma. What you're seeing here are little fragments from hotspots being connected one to the next in this kind of similar manner. So we call these tandem short template jumps. And the point here is that I want to emphasize that by single cell analysis back in 2015, we were able to identify a sequence signature whose significance we didn't know about, but we now have reason to think it's going to be widely important in cancer genomes. And although we don't understand the mechanism for generating this, where, I think I want to use this as an example of how this kind of mechanistic analysis may lead us to new mutational processes. And I have no data to support this, but just for your amusement, I want to just raise the fact that all of these little short templates are remarkably similar in size to Okazaki fragments. And Okazaki fragments, as you may know, are synthesized in a way so that there's partial overlap of the segments. So we're, considering models where these kind of rearrangements could have an origin from Okazaki fragments, but that'll have to be from for another talk. Okay, so I told you that bridges break. That leads to some complex fragmentation, which can lead and can also be associated with highly defective DNA replication that gives these very complex patterns. But then I'm going to show you that things get even worse. In the next mitosis, when the broken chromosome does something really surprising, the stub of the chromosome bridge that was poorly replicated undergoes massive DNA replication that occurs in a abnormal fashion as soon as the cell enters mitosis. So you get a burst of abnormal DNA replication. And that happens both on chromosome bridges and on micronuclear chromosomes. Here, because the images are most striking, I'm just going to show you the micronucleus, but we see the same result with chromosome bridges. So here, what we've done is to synchronize cells with micronuclei, where we have markers to show that the membrane on the micronucleus is intact in G2 with a CDK inhibitor. If we take these arrested cells and we pulse label them with EDU, we can see little, if any, DNA replication, little, if any, DNA damage. That was expected. And if we sequence these cells, only about 1 in 10 of the cells exhibits chromothripsis. So consistent with what I just showed you, there's a starting low level of chromothripsis that's probably associated with the bad DNA replication, but it's occurring at a very low frequency. However, now, if we release these cells and allow them to enter mitosis and then fix and stain them 20 minutes later, and here the EDU pulse is only happening during mitosis, you can see that there's a big burst of DNA replication here, and that's associated with a massive DNA damage signal. And that's real DNA damage because if we let cells go through mitosis and pick up the daughter cells and then sequence those, by contrast with the starting 10% level of frequency of chromothripsis, the frequency of chromothripsis is now jumping to about 90% in these cells that have passed through mitosis. Okay, and then things get even worse. I don't have time to show it to you, but the centromeres on the broken chromosomes appear to be inactivated. They appear to lose the SEMP-A epigenetic mark that specifies the chromosome, which means that in the next cell division cycle, those broken chromosomes again missegregate and become micronuclei, which could then generate additional rounds of chromothripsis. So we think that those are the main immediate consequences of the formation and breakage of the bridge. And if you believe that, what you would then say is that these events you could start with a single error in cell division and essentially rapidly recapitulate much of the complexity of a cancer genome just from that one event, because the initial events will predispose you to further bridging and rounds of chromothripsis. And to do that, we made chromosome bridges in a special way using CRISPR so that we knew that it was only chromosome 4 that was in the bridge. And we watched the bridges break and the two cells separate. And then we picked up these daughter cells, and instead of sequencing them, we let them grow up into populations and then evaluated what was going on in these populations either by sequencing, bulk sequencing of the entire population, extensive single cell DNA sequencing to look for heterogeneity in the populations, or growing subclones and directly doing bulk sequencing on those. And basically you can see here's the initial breakage site of the bridge, and you can kind of see the answer just from the copy number pattern of the two homologs here. If there was simple clonal alterations, you would have integer copy number alterations. You know, you would have 0 or 2 copies, for example, if the segment is gained or lost. But instead, what you can see is intermediate levels that are not integers. And that's telling you that the population is heterogeneous, and some have normal, some might have none, and some have at least double the amount of that chromosome. And in fact, if we clone to get subclones and then further sequencing those subclones, we can recapitulate the phylogeny of what went on. And again, this was recently published, and I'm just going to draw to your attention that we could see subclones that all shared one DNA breakage point, so they must have had a common ancestor. But the red lines here show you other breaks and rearrangements. And you can see that these other breaks and rearrangements must have occurred after the ancestral break. So again, the message is that a single error in cell division can drive a lot of the complexity of cancer genomes, including ongoing clonal subheterogeneity that is a major characteristic of tumor cells. Okay. So the take-homes for this part of my talk are that actomyosin contractile forces can interestingly promote mutagenesis. I didn't have time to show you preliminary data. This is not something that we've clearly yet established. That we believe that the stretching of chromatin in these bridges may also disrupt the centromeric epigenetic mark. And that's something that we're following up on. And that also has important consequences on the integrity of the chromosome. And I've shown you, I apologize, a rather complicated cascade of events that are shared by micronuclei and chromosome bridges that promotes chromothripsis and continuing genome evolution. And we'd like to suggest that this gives us a bit of a unifying understanding of the consequences of abnormal nuclear shape in cancer cells, which has been a long known feature of tumors. Finally, as I emphasized, a single error in cell division can generate a lot of complexity. I'd like to propose to you that a lot of the genome instability that we are observing in tumor cells is coming from abnormal nuclear architecture. For this point, for this part of the talk, I just want to highlight the outstanding work done by former postdoc Neil Umbright and former postdoc and now collaborator and assistant professor at Harvard Medical School, Cheng-Zhang Zhang, who has really spearheaded all the bioinformatic analysis. Okay, in the last few minutes, I just want to mention one practical application of this, which is that an important conclusion of the work that I just showed you is that one single DNA double-strand break that's not repaired has the capacity to break off a chromosome arm and generate a micronucleus. And if you had a replicated pair of sister chromatids that were both cut, you would then have the possibility of making chromosome bridges, which raised the possibility that Chromothripsis might inadvertently be a consequence of CRISPR-Cas9 mediated genome editing. And I'm just going to show a little bit of data from a paper that's on bioRxiv that shows that this is the case, which we think is at least a caveat that needs to be considered for human genome editing. Most of the work on concerns about genome editing have been on off-target effects of CRISPR-Cas9, which at least in principle can be addressed by improving the specificity of Cas9 or using other more specific nucleases. However, there's been growing evidence that there are on target, so that the site that is cut and edited can have additional abnormalities. And that includes small kilobase scale deletions and rearrangements near the cut site, megabase scale deletions that could be much of a chromosome arm, shown here in this study, and also complete loss of heterozygosity. And there's been a lot of debate about the origin of some of these on-target rearrangements, and there's no consensus about their cause. And I just I'm hoping that from what I just showed to you, that you might think that Chromothripsis might be one fairly obvious now cause that could explain some of these events. And as I said, that would occur because CRISPR-Cas9 will then create cuts, which could cut off an arm and lead to an acentric fragment that goes into a micronucleus. And this just shows that CRISPR-Cas9, including a clinically, a guide that's now in clinical trials here for sickle cell anemia, will induce appreciable rates of micronucleation. And this is done here in model cells, RPE1 cells. And we can verify that indeed this is cutting off the arm of the same chromosome, and we can verify by FISH that what we expect is happening is what is indeed happening. And if we sequence these cells, as expected, we see extensive chromothripsis. And I'm gonna, these plots actually underestimate, we're not showing all of the rearrangements. I wanna emphasize using very conservative bioinformatics. In this sample, we estimate that there are 646 chromosome rearrangements happening in one cell cycle after CRISPR-Cas9 genome editing in the cell that had the micronucleus. And again, following up on what I just showed you, if you have a replicated sister chromatid and you cut off the arm, you will now have two broken ends of the centric side of the break that can lead to chromosome bridges, as is illustrated here. And just like what I showed you here, I'm not going to drag you through all of the details. But we again could see examples where there were extremely complex events occur. You could see chromothripsis happening from a micronucleus. You could see evidence of the bridge forming that's also associated with chromothripsis. And because CRISPR-Cas9 can cut off acentric fragments like this that can float around and essentially be swapped, you can wind up with essentially the complete loss of heterozygosity happening also as a result CRISPR-Cas9 cutting because you're swapping acentric fragments of the chromosomes. Okay. And I'm just gonna, we're working with a close friend of mine, Mitch Weiss at St. Jude's Children's Hospital, where Mitch, along with many others, have now been working on a Cas9-based strategy to re-express fetal hemoglobin in human stem and progenitor cells so that you could complement the defect and treat sickle cell anemia by essentially losing an enhancer for a repressor of hemoglobin, fetal hemoglobin, and that repressor is called BCL11A. So if you don't have BCL11A, you make more fetal hemoglobin and fix the disease. And in primary human samples, in using the clinical, as best as we can, the clinical protocol, we do also get a low frequency of micronucleation and DNA damage. So, you know, this is a caveat, but, you know, not maybe more of a yellow light than a red light. I think this is something that the community needs to be aware of for human genome editing. We would suggest that we can monitor cells for the frequency of micronuclei. It may get easier as sequencing costs come down to actually directly monitor samples for Chromothripsis. For the sickle cell protocol, there's, I think, an important practical implication of this, which is that that protocol is essentially destroying the BCL11A enhancer by non-homologous end joining. And you do not need the cells to divide in order to get that editing event. So you could arrest the cells in G1 and get effective editing. And you would then, I think, very likely avoid the potential for Chromothripsis, because to get Chromothripsis, you need to divide and make either a micronucleus or a bridge. Obviously, many people are working on alternative editing strategies such as base editing or prime editing. Those are all interesting, but at an earlier stage and haven't advanced in clinical trials. I will mention, though, that all of these strategies themselves create DNA nicks, which can be converted into DNA double-stranded breaks. So these new strategies may help, but may not completely circumvent this problem. For this, I want to just highlight the work of two people in my lab, a graduate student, Mitchell Leibowitz, and a postdoc, Stamatis Papathanassiou, and all the human CD34 experiments were done with my friend Mitch Weiss. And there, I'll thank you for your attention and be happy to take questions.
Well, thank you, David, for a wonderful talk. It's really informative, and I personally have lots of questions myself, but we'll field the question from the audience first. Before you ask questions, just want to say that you can ask questions just by speaking directly, or you can type into the chat box. If you type in chat box, you can simply say, I have a question without detailing which question it is. The only thing I ask you to do is if you have a question in the chat box, we ask you to actually ask question in person so that we try to mimic the conventional feel of a seminar as much as possible. So maybe we'll start by one of the first questions asked in the chat is from Amitava Sinha. Maybe Amitava, you can start to ask questions. You actually have two. So why don't you go first and ask?
Hi, Professor. Can you hear me?
Yeah.
I have a question that while chromothripsis is going on, what is happening with mitochondrial function, as many of mitochondrial proteins are encoded, genomically encoded, and also if mitochondrial DNA is also getting changed and it is impacting somehow?
It's an interesting question. We have that hasn't been looked at to my knowledge. I mean, I would presume that most Chromothripsis events would be highly detrimental to cells. Most normal cells don't want you to blow up one of their chromosomes. And, you know, so you could, of course, compromise mitochondrial function. You can, you know, by copy number loss and rearrangement, also compromise, I presume, a variety of other cellular functions. However, the evidence that this can be, you know, allow mutations that cause increased growth in cancer is now incontrovertible. So rare. So even if most of the events are detrimental to cells, you know, it's essentially just a mutation. Most mutations are detrimental, but you can get rare ones under, you know, novel selective conditions that are advantageous.
So cells which are undergoing chromothripsis, so if they somehow overexpress DNA repair enzymes, can they survive?
Yeah, we haven't checked into that.
Okay. And these BFP cycles, while it is going on, do you think that there is any role of G quadruplexes?
Sure, and the telomeres, as you know, are rich in G-quad sequences, and that accounts for why they're difficult to replicate. And so I presume any defect in replication that's generally happening in the bridges might be amplified within the telomere repeats. I will say that in our sequencing, because we do throw out all repeat sequences, and that's actually something that we're kind of trying to look into in more general terms, is what's happening at the centromere and telomere repeats, but we haven't done that yet.
Okay, thank you.
Yeah, I see. The one thing I asked you, if you have a question that you're asking, please show your faces. That'll be the best. The next question is from the chat is Kai-Shuan Lin.
Thank you for your talk. I'm just wondering that once from bridge breaks, then what will happen to the telomere of the two cells? So if without the telomere, would the cell just go on forever from some rearrangement, or cells would die after several rounds of replication? Thank you so much.
Yeah, I mean, so I think that you raised an interesting, you know, I think there's an interesting and somewhat subtle point here, which is that one single double-stranded break might actually be worse than having 30 breaks. And the reason for that is that if you have 30 breaks in a mammalian cell, you cross the threshold where the DNA damage response can sense that there's a problem and will arrest the cell cycle and prevent division and allow time for repair. But if you only have a single break, it's not sensed, apparently. And then, so that's a problem. That break must be resolved. And it can either be resolved by de novo telomere addition, but that's very rare. So really the break has to be resolved by translocation. That's probably the major origin of repair for that. And if you don't repair the break, you're going to continue to generate more micronuclei and more bridges, which is why we think this is, you know, you're sliding down a slippery slope here, leading to massive genomic reorganization from one event and one break. Which is why, you know, which I think is the issue with CRISPR.
Thank you so much.
Andrew, you have a question?
Yes. Hi, David. This wonderful talk here. And so I have a question about the micronuclei's transcription status. Are they transcriptionally active?
Oh, well, I'm looking forward to talking to you. That was one of the reasons that I requested to chat with you.
Sure.
So they're highly, so we have done sort of adapted our procedures so that we can do single cell RNA sequencing. And when the micronuclei are intact, consistent with everything we've seen with DNA replication, we also see very strong defects in transcription.
Wow.
And when the micronucleus ruptures, then you seem to lose transcription. And then we've been following what happens in the next cell cycle to see whether there's memory, because you actually, the epigenetic marks on those cytoplasmic chromosomes get altered. And then if you look in the next cell division cycle, when they come into a normal, this is a work in progress and we don't have a complete answer yet, but we have preliminary results that there is some memory of this for the transcription. But the long and short of it is that I think these, maybe it's not really very surprising that, that everything is going wrong in these nuclear structures, including transcription, which might have some interesting implications for where transcriptional heterogeneity might be arising.
Okay, great, thanks. Look forward to talking to you.
Other questions? I think we've exhausted the questions listed in the chat, but if you have questions right now, you can just ask in the room. So maybe there I'll have a chance to ask my question here. So the chromosome bridges between the sort of dividing cells is really fascinating. I just wonder, is there any sort of a sequence or special properties of those DNA? Basically, any DNA can be there, or there's a preference of certain parts of the genome that will be preferentially be in those bridges?
We think any segment of the genome, there's, the bridges can either be between sister chromatids, so then you're going to have a bias towards the terminal segments probably being in the bridge more, but it's kind of, even that is kind of a subtle bias. And then with, you can have multiple chromosomes in the bridge from translocations, and they can be almost anywhere.
And do you have any idea what's the chromatin status of the DNA within the bridge? Apparently, they're not going to be the regular M-phase DNA. So also sort of coupled with the question that Andrew was asking, are there any evidence of transcription that's going on or absence of that going on in those bridges?
Yeah, so that's harder. So we haven't looked specifically at transcription, but again, what I've tried to convince you of is, or my theory is that bridges and micronuclei, although they look very different, are functionally probably the same thing. So I'm expecting that that would be the case. I mentioned, so if you take a chromosome in vitro and use an optical trap and pull on the chromosome at a force that's far lower than would be necessary to actually break the chromosome, you'll unspool the chromosome and you'll eject the histone octamers from the chromosome. And what preliminarily we see by live cell imaging is that when you have a bridge and the centromere sequence actually frequently gets trapped at the base of that bridge. And what we oftentimes see is that as we appear to see sort of localized contractile forces around the bridge, we appear to be able to see the loss of the centromere mark for the centromere Sambay. And we can see that being lost. And then when that's lost, we can follow the cells over another generation and see that that's functionally, essentially obliterated the kinetochore function, and that chromosome now behaves like an acentric chromosome arm. So if you can lose histones that are important for centromere function, I would presume that we think and are trying to test that you might also be able to lose chromatin marks that could affect transcription.
I see. Thank you. Megan has a question.
Hey, David, that was super cool. So just building on this idea, I was curious if you've looked at whether you actually get reversal of, for example, a mitotic H3 phosphorylation. In other words, do you actually get, what's happening to the condensation state, I guess, is the question. And I'm wondering if you've used things like ATAC-seq or something like this to look at the accessibility of the chromatin in the bridge.
Yeah, we're doing that, actually. Or at least I have a postdoc who's doing it. You definitely lose the H3K27 acetylation. That's like an extremely striking finding. And then we also see a large accumulation of H3K9 dimethylation. For some reason, we see more of the dimethylation than trimethylation. So we see stuff. We don't know what it means yet, but we are seeing stuff.
Any other questions from the audience? If not, let me propose that we give David a virtual round of applause and for a wonderful talk. Thank you, David.
Thank you.